All right, welcome back to another Explainer Grade 12 Life Science students. If you're gearing up for your final exams right now, well, you're exactly where you need to be. Today, we're unpacking the ultimate exam blueprint for one of the most heavily tested and honestly coolest topics in your whole syllabus. Okay, let's dive into this. We are mastering meiosis. I'm going to walk you through the entire shebang, the structures, the processes, why it matters, and even what happens when things go a little wrong. And we're doing it using the exact visual tools your examiners use. So, by the end of this, you'll be able to look at literally any cell division diagram and know precisely what's going on. Here's our quick roadmap for today. We're going to cover the importance of meiosis, walk through the actual process, compare the cell divisions, look at abnormal meiosis, and then wrap it all up with some real exam practice questions. Let's get right into it with part 1, the importance of meiosis, or as I like to call it, balancing the biology. Examiners absolutely love to ask why this process matters biologically.
So the crucial point is this. Meiosis is an absolute lifesaver because it overcomes the doubling effect of fertilization. I mean, think about it for a second. If human somatic cells have 46 chromosomes and a sperm and egg fused with 46 each, you'd end up with a baby that has 92 chromosomes. No way that works, right? So by producing haploid gametes, meaning they literally have half the standard chromosome number, meiosis perfectly balances out that doubling effect. It keeps the chromosome count constant from one generation to the very next. But wait, it doesn't stop there. Meiosis is basically the ultimate engine for genetic variation. Through this amazing mechanism called crossing over, plus the random arrangement of chromosomes right at the cell's equator, it guarantees that every single offspring is totally unique.
Moving on to part 2, the process of meiosis. This is where we visualize the divisions. Remember, visual identification is the most heavily tested skill here. Let's take a look at this beautiful overview. Now, before meiosis even begins, during a phase called interphase, the DNA actually replicates. But once meiosis officially kicks off, a diploid cell goes through 2 complete divisions. In the first division, meiosis 1, the number of cells doubles, but the number of chromosomes gets halved. Boom. Then, we jump straight into meiosis 2. And here, the chromosome number isn't reduced again. The cells just divide a second time. So what's the grand finale? We start with one single diploid cell and end up with 4 distinct, non-identical haploid gametes. Now, to nail this on an exam, I'm going to give you an infallible cheat code. Just remember 4 letters. P, M, A, T. You ready? P is for prophase, or preparation. The chromosomes condense, they become visible, and this is exactly where that crossing over takes place to bring about variation. M is for metaphase, or middle. The chromosomes line up perfectly at the middle, the equator of the cell. Then A is for anaphase, or apart. Chromosomes or chromatids are physically pulled apart by spindle fibers and move to opposite poles. Finally, T is for telophase, or terminal. This is the final phase where a new nuclear membrane forms and the cells physically divide. So P, M, A, T. Preparation, middle, apart, terminal. Burn that into your brain for sure. And it brilliantly illustrates exactly what PMAT looks like in action. Look at the left side, that's metaphase 1. You see those homologous chromosome pairs, the bivalents? They're lined up right across the middle equator of the cell. Notice how they are whole pairs. Then shift over to the right, stepping into anaphase 1, the spindle fibers contract, and whole entire chromosomes are being pulled apart toward the poles. They are not split chromatids yet, they are whole chromosomes moving away from the center. Being able to spot that whole chromosomes are separating, that's a massive, massive clue for your exams. All right, on to part 3, comparing cell divisions. Meiosis I versus meiosis II. Classic exam format coming up. Let's break this down together so you can completely dodge those common exam pitfalls. In meiosis I, chromosomes arrange at the equator in homologous pairs, but in meiosis II, they line up individually. During separation in anaphase I, whole chromosomes move to the opposite poles. But fast forward to anaphase II, the centromeres split and single daughter chromosomes, or chromatids, do the moving. On top of that, meiosis I halves the chromosome number, going from diploid to haploid, and crossing over definitely takes place. Meiosis II, on the other hand, just maintains that already halved number, and no crossing over happens there. So when you're staring at an exam diagram, just ask yourself, am I looking at pairs and whole chromosomes, or individuals and single chromatids? Hit the nail on the head with that question, and you'll spot the difference every single time. This brings us to part 4, abnormal meiosis, specifically understanding Down syndrome.
We've spent all this time looking at the delicate, perfect machinery of meiosis. But what happens when it breaks? This is a highly testable concept. The biological root cause of these chromosomal abnormalities basically boils down to one word: non-disjunction. Let's define this super clearly. Non-disjunction is the failure of homologous chromosomes or sister chromatids to separate properly during anaphase 1 or anaphase 2. So instead of moving apart equally to opposite poles like they're supposed to, they kind of stick together. This means one resulting cell gets too many chromosomes, and the other just doesn't get enough. The most frequently tested example of non-disjunction in humans is Down syndrome, otherwise known as trisomy 21. And here are the exact chronological steps you need to map out to get full marks on your exam. First, non-disjunction occurs at chromosome position 21 during anaphase. The pair simply fails to separate. Second, this glitch forms an abnormal gamete with an extra copy of chromosome 21, so it's holding 24 chromosomes instead of the usual 23. Third, if this abnormal gamete fuses with a totally normal gamete during fertilization, the math is straightforward. 24 plus 23 equals 47. The resulting zygote will have 47 chromosomes, specifically 3 copies of chromosome 21, and that condition is Down syndrome.
Okay, part 5, exam practice questions. Let's really test your knowledge. All right, we've covered the theory. Now it's time to put it to work. I want you to treat this next bit like an interactive exam simulator. When a question pops up, just mentally pause this explainer, look at the visual evidence, and actively try to recall the answers before I give them away. Now, what's really interesting about this slide is that these are exactly the type of diagrams you'll face. We've got diagram I on the left and diagram 2 on the right. Look at the spindle fibers, look at the equator, look at the poles. What phase are we actually in here? Are whole chromosomes involved or just chromatids? Scan the visuals and lock in your evidence.
Here's your first mock question. Based on diagram I, state one visible reason indicating that meiosis is taking place, and then describe what takes place in the cell right after the phase shown in diagram I. I'll give you a quick second to scan the image. Look really closely at what's happening to the chromosomes as they separate. All right, here's the memo. If you said that whole homologous chromosomes are separating, you are absolutely correct and you just scored the mark. Because they are whole chromosomes and not just chromatids, we know for a fact this is anaphase 1. Now, the second part of the question asked what happens next. Well, remembering our trusty PMAT cheat code, after anaphase comes telophase. So telophase 1 occurs, a nuclear membrane forms, and cytokinesis kicks in to split the mother cell into 2 daughter cells. Great job validating your active recall. Let's move to our final practice question and see how this builds on our abnormality knowledge. Here's the scenario. If an abnormal egg cell with 24 chromosomes fuses with a normal sperm cell, name and explain the resulting disorder. Think back to those precise steps of non-disjunction we covered just a few minutes ago. Okay, here's exactly how you unpack this for the full 3 marks on the grading rubric. First, you have to name the disorder, Down syndrome or trisomy 21. Then you explain the math. The abnormal egg cell has an extra chromosome, meaning it has 2 copies of chromosome 21. It then fuses with a normal sperm cell that has the standard 1 copy of chromosome 21. Therefore, the resulting zygote will end up with 3 copies of chromosome 21. Nailing that specific phrasing about the final zygote having 3 copies is exactly what the examiners are hunting for. And just like that, we've reached the end of our meiosis masterclass. It's pretty wild to think about, but meiosis literally guarantees our genetic uniqueness. Every single one of you watching this explainer right now is completely unique because of the crossing over and random arrangement that happened in your parents' biology before you were even a thought. So, based on what you've revised with us today, if I showed you a slide right now, could you spot the exact prophase phi and metaphase phi moments where your unique traits were decided? I bet you totally could. Keep practicing with these visuals, trust your PMAT structure, and you're going to absolutely crush your life sciences exams. Thanks so much for joining me on this explainer, and keep learning.